So in this video, I'm going to show you how to do a gene sequence alignment using Bioedit software. So make sure that you have already downloaded this Bioedit sequence alignment editor before proceeding and finding your sequence of interest. And the first step that we have to do when we want to align a gene sequence using the Bioedit is to have the sequence in FASTA format. So now we are at the NCBI website to download a gene sequence. So you just need to type on the gene of interest that you would like to have. So for me, it's the hantavirus L99. And make sure that on the drop-down menu, you would select on to nucleotide and then search on it. Okay, so in this area, you would see a lot of options, around 5 options, depending on your query. So we will choose around 3 sequences for this, and we will download it using the FASTA format. So I have chosen numbers 1, 3, and 5. And now let's go to each page. And let's wait for it to load. So the first one is the hantavirus L99 segment M envelope glycoprotein mRNA complete CDS. So as you can see here, it has CDS. So this stands for coding sequence. So however, this will not contain if any information regarding introns. So that's what it's all about. So what can you see here in this page is just a typical nucleotide database. So the accession number, the version of it, the organism, the reference, the authors, the title, and a lot more, as well as the sequence. So now to download this, we just need to click Send to, File, and then we will choose the format. So I would like to use the FASTA format or the FASTA format for our Bioedit. So let's just create the file and it will be downloaded on this one. Or you can just simply click on this part, FASTA area, and it will take you to the downloading side or page of this sequence. Next on is the hantavirus L99 nucleocapsid protein gene complete CDS, just like what we have earlier. So just as the same, the accession number, the keywords, organism, reference, author, and now let's download it. Once again, Send to, File, FASTA format, Create File. So it will then be downloaded again. And next one, or the last one, is the hantavirus L99 segment S nucleocapsid protein mRNA complete CDS. So this time we will use this FASTA format button on the left hand side. So once you click this, it will take you to the page of the sequence. So you will also click Send to, File. So it's already in the FASTA format. So this is just to show you that once you click the FASTA format, it will take you to a page where the sequence is only there, because other people might be confused to find the sequences if they are not familiarized with this website. So once again, send to File, always check the format and then create the file. So we already have 3 sequences, and now let's head on over to the Bioedit sequence editor. So first, let's familiarize ourselves from the toolbar of this software. So for the file, we would have this new alignment, open, new from clipboard, open as text, a lot more. While on the accessory application, you can see here the blast, the Clustal W example application, which is very important later because we will use it in the alignment of the sequences. The DNA distance matrix, this prot-parse protein parsimony method, the RNA matrix plotter, the World Wide Web, options, window, and help. These 2 icons are for open sequence set and new alignment. So we can input multiple sequences or sequence files into one window. So now let's open File and then New Alignment. Or we can just use the combination Ctrl+N. Now to add the sequences, let's click on File and then Import. Next is the sequence alignment file. So now let's just change it to all files, and then we can see here the sequences that we have downloaded a while ago. So let's just click open, and there you go. Once all the sequence has been selected, so we can already run Clustal W from the accessory application. So let's click on Clustal W multiple alignment, run Clustal W, then click OK. So once it is done, let's click on Alignment, then find Conserve Regions. So then let's click on Start. So no regions were found that conform to the given parameters. So this result just shows us that there are no conserved regions found. So if you encounter sequences that has conserved regions, you can use this for real-time PCR and probe design. It will serve as a template for that. So you would just need to check its specificity by the region, and you could use the nucleotide blast in this software. So the conserved regions are important because these regions will have least mutations. So these are ideal regions for designing primers and probe for real-time PCR. So that is how you align gene sequence using the Bioedit Sequence Alignment Editor.